Hello. Good afternoon. We'd like to welcome all of you to this session on insights into initiation and progression of PDAC, advances of early detection of PDAC. I'm Ajay Goyal from City of Hope Cancer Center in LA, and my co-moderator, Dr. Laura Wood from Hopkins. So we'd like to start off, we have 4 talks in this session, but unfortunately, 2 of the speakers couldn't make it in person. So we're gonna go, first 2 speakers are present and the other 2 will be virtual. So we'll start off with the first talk by Dr. Wood, and she'll be talking about overview of pancreatic cancer diagnosis, early detection, and genomic sequencing.
Thank you so much to the organizers for the invitation to speak. And I have this very large task and very broad topic in 12 minutes to get through it. Thankfully, we had some amazing symposia yesterday that covered some of the ideas in the talk. So when we think about diagnosis of and early detection, Dr. Simeone nicely highlighted yesterday that most patients don't develop symptoms until advanced stage. And so we need early detection to allow intervention when resection can be curative. There are 2 key components of early detection programs, and that is whom to screen and then how do we do the screening. We talk about whom to screen, and again, I hope everybody was able to hear the Hirschberg Symposium yesterday, which highlighted a lot of these ideas. But population screening in pancreatic cancer is not viable because of the relatively low incidence, right? You'd need, even with a very, very good test, you have too many false positives. So you need to enrich your population for high-risk individuals who are most likely to benefit from screening. And we know what some of these populations are, right? Folks with family history and germline alterations, as discussed by Dr. Canto, pancreatic cysts, folks with new onset diabetes, as discussed by Dr. Chari yesterday. So we know a lot about whom to screen, but then how do we do the screening? Again, in the Hirschberg Symposium yesterday, we heard a lot about radiographic imaging, which can identify lesions in high-risk individuals, but many of these lesions are low risk and are not going to progress. And so we need additional tools to risk stratify these lesions for which ones are most likely to progress and which ones should be intervened on surgically. We heard some great data yesterday on imaging features and radiomics and using those tools to risk stratify. But what I want to talk more about are biomarkers for the same task. And the third part of my task was to talk about genomics. So as you might suspect, I want to talk about whether genomic alteration can serve as these biomarkers to stratify low risk from high risk, lesions at low risk from high risk for progression. So it's been 14 years now since we got our first comprehensive look at the genomic landscape of pancreatic cancer. This is data from Bert Vogelstein's group, who did the first whole exome sequencing of human pancreatic cancer. This was 24 pancreatic cancer cell lines that were analyzed by whole exome sequencing. And what that analysis showed is that there's 4 huge genomic mountains that are altered in the majority of pancreatic cancer patients. These are the oncogene KRAS, and then 3 tumor suppressor genes, which are P16 or CDKN2A, SMAD4, and TP53. But the landscape is also filled with these hills that are less frequently altered, but they're still driving pancreatic tumorigenesis. In those 14 years, we've now sequenced hundreds and hundreds and hundreds of pancreatic cancers with whole exome or whole genome sequencing, both through large coordinated efforts like the Cancer Genome Atlas, the International Cancer Genome Consortium, as well as large institutional groups. But not surprisingly, the most frequently altered genes are still the 4 mountains, right? It's KRAS, P53, P16, and SMAD4. But It has also really clarified what the hills are, some important functional groups of genes that are also altered. We heard yesterday from Dr. O'Reilly about the DNA damage repair genes and how those are important clinically for response to therapy. But our task today is not therapy, it's early detection. And so how can genomics impact early detection? We need to directly analyze the precursor lesions to understand how they evolve and how we can apply genomic principles to these lesions. There are two different flavors of pancreatic cancer precursor lesions. So most pancreatic cancers come from microscopic pancreatic intraepithelial neoplasia, or PANN. These are by definition less than 5 millimeters, meaning they're challenging to detect radiographically, and they're graded based on the morphologic dysplasia of their epithelium. Low-grade PANNs have mild to moderate atypia. They're very, very common. High-grade PANNs, much more severe atypia morphologically, and the next step would be transformation to invasive carcinoma. Intraductal papillary mucus neoplasms, or IPMNs. These are large cystic lesions, by definition greater than 1 centimeter. They're often much larger than that. These are detectable radiographically. They're often discovered incidentally, and this leads to a clinical challenge of who do we surveil and who do we intervene on surgically? And that's one space in which molecular biomarkers can have some use. So my group has done a fair bit of work analyzing human tissue samples to really understand clonal evolution in IPMNs. I've presented this data at APA before, so I won't go into details, but rather just highlight broad concepts, which is that unlike advanced pancreatic cancer that are often genetically quite homogeneous, there's a lot of genetic heterogeneity with respect to these driver genes in the precancers. They often have a polyclonal origin, so there's 2 distinct clones marked by different KRAS mutations, and then heterogeneity with respect to the other driver genes as they progress before there's a single clone that transforms to invasive cancer, and then you have this cancer that's homogeneous with respect to the driver genes. And again, just thinking of this progression, right? When you start with normal epithelium, a low-grade IPMN has multiple KRAS mutations. These KRAS oncogenic mutations are really the earliest alteration. And then you get multiple rounds of selection and eventually invasion into pancreatic cancer. So can genetic alterations identify high-risk IPMNs? This is data from a recent paper from Ator Singh's group at Pittsburgh. There's also similar efforts at Hopkins to sequence IPMNs this fluid, look at the genetic alterations, and see how that correlates with grade of dysplasia. And I want to highlight here, these are IPMNs with low-grade dysplasia, IPMNs with high-grade dysplasia or cancer. If you look at KRAS and the other oncogene GNAS, the patterns are quite similar between the two. These are early alterations that don't differ between low grade and high grade. Where you see differences are in these later alterations like P53 and SMAD4, and you can see a lot of blue lines for mutations in the high grade and very few in the low grade. So can we use these later driver gene mutations to identify high-risk IPMNs? We recently did a systematic review of this question, and what we found is that these later driver gene mutations, the sensitivity of them for high-grade IPMN is not good. It's quite low, but the specificity is really high. So they're 95 to 100% specific for either high-grade dysplasia or cancer. So this suggests that if you see an alteration in one of these late driver genes, the patient is very likely to have either high-grade dysplasia or cancer, but a lack of these mutations doesn't exclude it. Again, the sensitivity is low, so we need new biomarkers. But what about PANNs? The majority of pancreatic cancers arise through the PANN pathway. What do we know about the genetic alterations in PANNs? So PANNs are microscopic, so they're much more challenging to study. So to get at this, we collaborated with Denis Wirtz and Pei-Shun Wu, who are engineers at Hopkins, their student Ashley Keeman, and my graduate student Alicia Braxton, and we developed a way to 3D model pancreatic tissue so that we could quantify PANN and then do genomic mapping. So this 3D reconstruction approach is called CODA. What we do is we take a large piece of human pancreatic tissue, we serially section it, and we do H&E staining of all the slides, and then we can scan them and do image registration. We've trained a deep learning algorithm that can identify all the tissue types, and then we can do cellular detection and quantification, and it lets us visualize these pieces of pancreas in 3 dimensions and then quantify all of the cell types. This video did not work on the test, and it looks like it is not going to work this time. So I will just kind of explain what it showed. there we go. So this is a large piece of human pancreas. It's about 2 cubic centimeters, and it has about 1 billion cells in it. And what CODA lets us do, I wish the video was running, but it lets us determine where all of the cell types are, what their relationships are to each other, and we can go down to the single cell level. The next video I'm hoping will work. And this is one that's going to show different pannins. And so the normal duct is going to be in blue, and each color in the figure is going to be an anatomically separate pannin. And so when we looked at these, we were quite shocked with kind of the number and density of pannins. Again, this is about 2 cubic centimeters, and there's more than 10 pannins in this one fairly small piece of pancreas. And this is zooming in on one of those anatomically separate pannins. And again, this 3D modeling lets us make these nice videos, but also lets us quantify all of the cells and all of the relationships to the pannins and the other cells in the tissue. So we've now modeled 38 pieces of grossly normal pancreas. These were all from surgical resections. About half had pancreatic adenocarcinoma, the other half had other diseases in their pancreas. And we found across the whole cohort a really shocking burden of pannin. This shows, I think, 15 of the models. And you can see that all of them have multiple spatially independent pannins. The average in our cohort was 24 pannins in each piece of tissue that was about 2 cubic centimeters. So kind of a shockingly high pannin burden. The vast majority were low grade. Of the 889 pannins that we modeled, only 3 were high grade. We use this to calculate the potential pannin burden because we know the size of the pieces of tissue we harvested, and we also know the average size of a pancreas based on a large cohort of CT scans. And when we do this extrapolation for both men and women, the mean number from our cohort to extrapolate to the entire pancreas would be over 700 pannins. So it's a huge number of pannins that are occurring grossly normal pancreas. One of the questions we had, we have this large number of pannins, but are they each independent neoplasms, or is it one neoplasm that's traveling through the duct system? And to get at this question, we did genomic analysis of these pannins. We took a separate cohort of 8 patients. When we did the sectioning, we sectioned some onto membrane slides and then microdissected the individual pannins, and then separately analyzed them. And this is one example where this patient had 4 different pannins as well as a PDAC. And you can see in each pannin, they had a different KRAS mutation. This suggests that they're all separate neoplasms, but we wanted to confirm it with exome sequencing, which we did. And we saw that there was no overlap in the somatic mutations in the different pannins. These 2 that overlap are actually the low grade and high grade component of the same pannin. So that's the only large overlap of somatic mutations. We also found some pannins that had multiple different KRAS mutations within the same spatially contiguous pannin. You'll just look at the yellow pannin for an example. And here are the mutations. You can see that this one pannin that's spatially contiguous has 6 different KRAS mutations. We also confirmed these with an ultra deep KRAS sequencing technique, really showing that the themes that emerge in IPMNs with multiple KRAS mutations and polyclonal origin probably also apply to pannins. So the take-home points in this 3D pannin analysis, we have these large number of spatially separate pannins that occur within a relatively small sample of pancreas. Most of these multifocal pannins arise from independent genetic events. We had a couple examples of intraductal spread, but it's really quite uncommon. And some have multiple KRAS mutations suggesting a polyclonal origin. This is our kind of first look. There's a lot of important future directions. One is the mechanism for this multifocal neoplasia, and I think our next speaker will touch on some of those themes. As I mentioned, our cohort was largely low grade. So what are the alterations that are associated with progression to high grade? We need a dedicated analysis of high grade pannins. And then finally, all of these patients were surgical patients. Not all of them had PDAC, but they all had a mass. And so we need to look at truly normal pancreas across the age spectrum. So just finally, I think I've hit 12 minutes. So it's a huge topic to do in this time, but we need early detection strategies to improve survival of pancreatic cancer patients. We need to know whom to screen and how to screen. I think the genetic alterations in the late driver gene mutations are quite specific for IPMN risk stratification, but they're not sensitive. We need new markers as well. And then finally, these molecular strategies for pannin risk stratification are going to be challenging just because there are so many pannins. We have such a high pannin burden. And with that, I want to briefly acknowledge the folks in my lab and then our funding, and then I'm happy to take questions. Thank you. Oh, we're going to do questions at the end. Sorry. I'm feeling my job as moderator. And now it's my pleasure to introduce our next speaker, who is Dr. Andrea Viale from MD Anderson, who is an associate professor in the Department of Genomic Medicine and a faculty member in the Graduate School of Biomedical Sciences.
Good afternoon, everyone. Thank you, the organizer, for the invitation and the opportunity to present our recent work about tissue damage, inflammation, and pancreatic cancer. I do not have any disclosure beside an international patent about ideas that actually I'm going to discuss at the end of this talk. So when we talk about, let's say that pancreatic cancer progression is being considered a stepwise process characterized by the accumulation of defined genetic alteration and also histological alteration, although as demonstrated, the progression is a process that actually is for sure much more complex. Specifically here, we are going to talk about a very early lesion that actually is ADM, so the acinoductal metaplasia. So when we discuss about tumor progression in pancreatic cancer, we have two major players that actually are big elephant in the room. One is KRAS, that is virtually mutated in all the patients. And so the big question here is why? And the second thing also is obviously inflammation. Okay, so we know that pancreatitis is a major risk factor for pancreatic cancer. We know that actually a large part of patients apparently does not have an history of pancreatitis, but there is no question that inflammation hasten tumor development in mouse model. One of the things that actually, as cancer biologists, we forget when we study inflammation and cancer is the fact that inflammation evolved as a non-specific damage response to coordinate activation of the innate immunity and tissue repair. And so based on, with this concept in mind, two trainees in my group were interested in really studying the effect of inflammation in normal epithelial cells. And for doing this, basically, we just expose animals to a very short inflammatory, a very short inflammation, a couple of days. We, on purpose, didn't want to have a longer protocol, use a long protocol for chronic inflammation because we didn't want to have any alteration, okay, at the level of the stroma. That could be confounding. So basically, after 2 days of inflammation, obviously the pancreas recover perfectly. By day 7, histological level, we didn't have any. So the pancreas was normal. As the molecular level, we have the completely extinguished. So the molecularly, the inflammation was completely extinguished. But when we characterize better the epithelial cells, we found that actually the epithelial cells recover from a transient inflammation through a sustained gene expression and chromatin reprogramming that we name as epithelial memory. So you can clearly see here that specifically epithelial cells and actually specifically acinar cells, not ductal cells, not other cells in the stroma and no endocrine cells, basically they get, they undergo a drastic transcriptional deregulation after inflammation at day 1, so the pink color there. And then the cells, they move back towards the blue, the basal level, but they never reach the basal transcriptional state. They get stuck somewhere in the middle, okay, in a new transcriptional state that is also mirrored by modification at the chromatin level, as you can appreciate here from ATAC-seq. So basically, we had the activation of programs that usually are activated during ontogenesis and also reactivated during tumor progression. So at that point, we try to activate KRAS And what we found is that basically after 1 month of the recovery, in the animals that actually were exposed to pre-exposed to inflammation, basically the animal develop PDAC with a very high penetrance and with a very high penetrance. And this is true also when we activated KRAS several months after the resolution of the inflammation. We activated until after 3 months, and still we got the same phenotype. And interestingly, we got the same phenotype using other models of inflammation like ligation. And interestingly, the penetrance and is really similar, and the latency is really similar to what actually we get when we have an active inflammatory event ongoing. So we spent time to characterize at the molecular level what actually was happening. We identify IL-6 as one of the major mediator, and we identify also transcriptional factors that can be responsible for the reprogramming. But what I would like to discuss today is really about the adaptation itself, and which are the advantages that are conferred to pancreatic cells by epithelial memory. So to test, so to explore what was going on, basically we just re-challenge the animals that were previously exposed to one inflammation with a second inflammation. And as you can see here, in terms of enzyme in the peripheral blood or assessment of damage, it looks like that actually the animals with memory basically process the same stimulus in a milder way. Okay, we had a milder pancreatitis. And what actually was interesting is that the animals with memory undergo the second inflammation, activating in a very fast way ADM, okay, metaplastic lesion. So we started really to think about that the metaplastic lesion could really have some role in limiting tissue damage. And it makes sense, right? Because I mean, when acinar cells shut down the acinar program to become similar to ductal cells, obviously they stop to produce enzyme that if they get activated can really fuel inflammation. And since actually this process has been known to be mediated by activation of MAP kinases, what we tried to do here was really to play with the activation of MAP kinase signaling and modulate the signaling using EGF as an activator or a MEK inhibitor as obviously an inhibitor. And what we found is that in presence of EGF, we got an expansion of the ADM and a complete reduction of the damage in the tissue. The scale here is logarithmic. That is mirrored also by the LDH level in the peripheral blood. So just demonstrating that really maybe metaplastic lesion really can protect the tissue from the damage from inflammation. So then the next question was quite obvious, right? So I mean, which is the role of KRAS? KRAS is upstream MAP kinases, and so we really try to test the effect of oncogenic KRAS induction on inflammation. And what we found is to use a genetic model to activate KRAS specifically 1 day before the second inflammation. And what we got at that point was really the induction of very massive ADM and a complete suppression of damage in the organ. So these data are important because we think actually, give us a different perspective of progression of PDAC. So we were able to demonstrate that the epithelial memory facilitating really the rapid acquisition of metaplastic lesion evolved probably as a protective mechanism to limit pancreatic damage and support tissue repair. And the other thing is that really the stabilization of prenoplastic lesion by activating mutation of KRAS make KRAS, mutated KRAS, oncogenic KRAS, a beneficial event in recurrent pancreatitis. So uncovering probably, let me say, a physiological role of oncogenic mutation in preserving the tissue integrity during damage. So we speculate that really probably due to this protective effect of oncogenic KRAS, the mutation can be selected in non-neoplastic tissue early on and independently from tumorigenesis. And at this point can really explain why some oncogenes, specifically KRAS in pancreatic cancer, basically are a universal event. Let me be at this point a little bit provocative. So everyone wants to inhibit things. We actually want to do exactly the opposite. And so if early step of tumorigenesis represent an adaptation to perturbation of tissue homeostasis, can we use pharmacological modulation of preneoplastic lesion to ameliorate pancreatitis and halt tumor progression? So Ruby, a student in my group, is working on this project. We identified a couple of classes of drug already approved by FDA or in late clinical trial that actually are able to induce metaplastic lesions. So the point was really trying to test the hypothesis that if we induce metaplastic lesion, if we can shut down inflammation and in the same time really decreasing the pressure to mutate the KRAS. So I'm going to show you some preliminary data about inflammation. Is too early for mutation of KRAS. That is a very complicated study. So one of the class is represented by BET inhibitors, and they work really well. So what we do not know exactly how. So we are working on this. We have obviously idea. We can discuss later on. But as you can see here, so basically we treated the animals with BET inhibitor at the time of inflammation. BET inhibitors are not doing anything on the normal pancreas, but together with inflammation, they induce very beautiful ADM. And we have a complete suppression of infiltration of CD45 in the tissue. And as you can see here from the quantification, okay, so the BET inhibitor are better than the previous biologics that we use, like EGF, and are much, much better than a conventional anti-inflammatory drug. So in summary, really, we start to think about that pancreatic tumorigenesis is in reality an unintended consequence of an early tissue adaptation to inflammatory damage that leads to the fixation and selection of KRAS mutation because of the protective role in preserving tissue integrity. Okay, this can explain why KRAS is a universal driver. At the same time, we really think that actually the reversible induction of metaplastic lesion can be exploited pharmacologically as a novel effective means to suppress inflammation, limit damage, and promote tissue repair during pancreatitis, representing probably one of the first physiological method of intervention. So really, we are not inventing anything. Here, we are just basically, mimicking what actually nature is doing. We do not have at this time data on prevention of pancreatic cancer. We are working actively on this. So thank you for your time. I'm running late, and yeah, I can just keep the acknowledgement. Okay. Thank you.
Thank you very much. So the next talk is going to be by Dr. Mandar Majumdar from Yale University School of Medicine. So this is a pre-recorded talk, and you will hear, he'll join us at the end of the talk for questions and answers.
I want to thank the organizers for putting together a wonderful meeting and for the exciting opportunity to speak about our work today exploring the mechanistic links between diet, obesity, and pancreatic cancer. My name is Mandar Mazumdar, and I'm an assistant professor in the Department of Genetics and a core member of the Cancer Biology Institute at Yale University. I apologize that I cannot be with you in person today, but I look forward to your questions and comments in the live panel discussion. My lab is broadly interested in the genetic, environmental, and host factors that drive cancer progression, with a special focus on pancreatic ductal adenocarcinoma, or PDAC, a highly lethal cancer with a 5-year survival rate of approximately 11%. One such modifiable host factor, obesity, is a major risk factor for pancreatic cancer, and over a dozen other cancer types, including nearly all cancers originating from the gastrointestinal tract. As you know, the prevalence of obesity has risen steadily over the last 5 decades worldwide, and at a much more alarming rate in the United States. As a result, obesity is thought to be a major contributor to the overall rising incidence of pancreatic cancer globally. Therefore, there's a desperate need to better understand the mechanisms by which obesity drives the development of pancreatic cancer, to develop novel strategies for prevention or treatment for this highly recalcitrant cancer. To determine whether obesity plays a causal role in PDAC development, we crossed the KC mouse, an oncogenic KRAS-driven model that closely recapitulates the genetic and histologic features of human PDAC progression, with a widely used genetic model of obesity, the OB-OB mouse. OB-OB mice are deficient in the satiety hormone leptin, leading to rapid onset obesity on normal mouse chow to more than 2-fold the body weight of their leptin intact littermates within 2 months of age, as shown here. Resultant KCO mice exhibited a significant increase in tumor development and a marked reduction in median survival, consistent with obesity as a driver of pancreatic tumorigenesis. The OB-OB model gave us a tractable tool to test whether reversion of obesity can intercept tumorigenesis, as it is susceptible to rapid weight loss either via leptin replacement, which we performed using an AAV-based genetic approach, or dietary caloric restriction. Strikingly, we found that early but not late weight loss intercepted tumor development to near baseline levels, arguing that obesity plays a causal and reversible role in early PDAC progression. Thus far, I have shown you that excessive food intake and resultant obesity can drive pancreatic tumorigenesis. Perhaps a question of equal, if not more, translational relevance is, does what we eat matter as much as how much we eat? For the remainder of the talk, I will focus on dietary fat, as the modern Western diet is characterized in part by high levels of calories from fat. In fact, total fat consumption has been on the rise over the last half century in the United States and worldwide, mirroring the ascent in obesity rates I had shown you earlier. This has particularly been driven by increased consumption of a variety of vegetable oils, as opposed to the lard-based, high-fat diets used in most preclinical cancer research. Core studies exploring a link between fat consumption and PDAC risk have shown more consistent results than obesity studies using objective measures, such as body mass index. Indeed, these nutritional epidemiology studies are confounded by dietary recall bias, lack of granularity in diet surveys, and a small sample size. Therefore, whether and how specific dietary fat sources and fatty acids play a role in pancreatic tumorigenesis remains poorly understood. To address this question, we teamed up with Dr. Matthew Rodehafer at Yale, who developed a panel of 12 isocaloric high-fat diets, differing solely in fat source and representing the diversity of modern fat consumption. The panel included lard, the most commonly used fat source in cancer obesity studies, but also a variety of vegetable oils that are more commonly consumed by humans today. Importantly, the high-fat diets diverge significantly in the proportion of different fatty acids, enabling us to match dietary effects on tumorigenesis to specific fatty acid components. We fed this high-fat diet panel to congenic KC mice for 12 weeks and analyzed tumor development. Importantly, no major differences in overall food intake were observed across the diets. We found that dietary fat source dictated the level of tumor burden observed in these mice. Few mice developed PDAC within this time frame. Thus, the phenotypic effects were largely limited to formation of early tumors, including pre-invasive pancreatic intraepithelial neoplasia, or PINs. Strikingly, the 3 most tumor-promoting high-fat diets, olive oil, high oleic safflower, and high oleic sunflower oil, had the highest concentrations of oleic acid, a monounsaturated fat typically associated with improved cardiovascular health. Serendipitously, our diet panel contained non-OA supplemented versions of the high oleic safflower and sunflower oils, allowing us to directly compare how OA concentration or oleic acid concentration affects tumorigenesis by controlling for the effects of these base diets. Indeed, we observed that oleic acid supplementation was sufficient to enhance tumor development across both these diet pairs. Most of our mature data thus far examining the relationship between fatty acids and cancer has been derived from male KC mice. However, our early studies in female KC mice confirmed that high oleic oils such as olive oil also promote tumorigenesis, in contrast to traditional large-based diets. And we have ongoing studies to determine if this remains true across our diet pan. In further support of a role for dietary oleic acid in tumor formation, we saw a positive correlation between both dietary and total plasma oleic acid levels and median tumor burden across our entire diet panel. These findings mirror the positive correlation observed between human per capita oleic acid consumption and the risk of developing PDAC, where each dot on this graph represents a different country, using available data from the WHO and Global Cancer Observatory. Together, these data argue that excess dietary oleic acid consumption may be a driver of PDAC development in mice and humans. But how does oleic acid function to promote pancreatic tumorigenesis? One hypothesis is that excess dietary oleic acid induces inflammation, a known risk factor for PDAC development. Contrary to this hypothesis, we did not observe histologic differences in the level of inflammation or fibrosis induced by high-fat diets low or high in oleic acid. Instead, we found evidence that oleic acid could directly induce changes in acinar cell identity that may facilitate tumorigenesis. RNA sequencing analysis of bulk pancreata show loss of expression of acinar cell identity genes and acquisition of duct marker expression in KC mice fed high oleic diets. We further found that oleic acid could directly induce the expression of duct markers at both the RNA and protein level in primary acinar cells, arguing that oleic acid might promote acinar ductal metaplasia, a prerequisite step in early PDAC development. Given the direct phenotypic effects of oleic acid on acinar cells, we postulated that oleic acid might directly incorporate into cellular lipids within the pancreas to exert its pro-tumorigenic effects. In support of this hypothesis, we found that tissue incorporation of oleic acid into fatty acyl chains of specific membrane and cytoplasmic phospholipids, including phosphatidylcholine, phosphatidylethanolamin, and their hydrolyzed lysophospholipid forms, LPC and LPE, strongly correlated with tumor development. Tissue levels of these 18-1 oleic acid containing phospholipids, here shown for the liver, were equal to or even more strongly correlated with tumor development than circulating OA levels, arguing that oleic acid incorporation into bioactive cellular phospholipids may be critical for its ability to promote tumor formation. Indeed, we observed specific enrichment of these very same oleic acid containing phospholipids in the pancreas of KC mice fed oleic acid supplemented diets. Beyond acquisition from the diet, oleic acid can also be endogenously produced through de novo lipogenesis, or DNL. We found that key biosynthetic enzymes involved in DNL were upregulated in pancreata of mice fed high oleic diets, as were phospholipid remodeling enzymes involved in incorporating exogenous oleic acid into the acyl chains of cellular phospholipids. We further observed that oleic acid could directly induce these DNL genes in primary acinar cells and functionally promote de novo lipogenesis in PDAC cells in delipidated media, shown here by the increase in palmitate, an early product of DNL. Thus, exogenous oleic acid increases available cellular oleic acid levels not only through its uptake, but also through induction of its own synthesis in pancreatic cells. Based on these data, our working model is that excess dietary oleic acid is directly taken up by tumor-initiating acinar cells from the pancreas. It is incorporated into specific phospholipid species to reprogram cellular identity and lipid metabolism to drive tumorigenesis. This model leads to the hypothesis that reducing cellular oleic acid levels in the pancreas may suppress tumorigenesis. We're currently using a variety of genetic, pharmacologic, and isotope tracing approaches to test this hypothesis by decreasing oleic acid availability in the diet, inhibiting endogenous synthesis, blocking its cellular uptake in pancreatic cells, and inhibiting the remodeling process that leads to its incorporation into bioactive cellular phospholipids. Together, these studies may delineate key targetable pathways to subvert oleic acid-driven pancreatic tumorigenesis. During the course of the talk, I hope I've convinced you that when it comes to diet and pancreatic cancer, both quantity and quality matter. That there's a tremendous value in pursuing systematic controlled diet studies in faithful in vivo cancer models, as unexpected relationships may be identified, which are not easily gleaned from nutritional epidemiology studies due to confounding variables. And that specific dietary fatty acids may play direct roles in tumorigenesis, arguing for more expansive diet panels when studying the relationships between diet, obesity, and cancer in preclinical models. Finally, our data suggests that there may not be a one-size-fits-all approach to nutritional guidance. Whereas oleic acid may be beneficial for those at high risk for cardiovascular disease, perhaps not so for those at high risk for PDAC. With that, I want to thank the many members of the lab who contributed to this work, especially postdoc Christian Ruiz, who led the project, and a postgraduate, Riley McDonald, who worked closely with Christian on the project. We're indebted to collaborators at Yale and beyond, and to multiple sources of critical funding for this work. Thanks.
Our final speaker will also be delivering a recorded talk, but will join us for the Q&A. This is Dr. Akihisa Fukuda, who is an associate professor at Kyoto University and will be talking about PDAC imaging.
Hello, everyone. Thank you for the great opportunity of this presentation today. I'd like to talk about visualization of PDAC stem cells by lineage tracing. Hello. As a candidate for a marker of pancreatic cancer stem cells, we focused on DCLK1.
Please bear with us. Sorry, I cannot hear you. Okay.
Hi. Hello.
Dr. Fukuda, can you please talk while we...
Okay.
Thank you.
Just a moment, please.
Thank you. Dr. Fukuda, we can't hear you. Seems like you. So can you please,
Can you hear me?
We can hear you, but your recorded presentation is not working. So if you can please, we can present the slides for you if you would like to speak.
Okay. Can I present by? Can I share my slides?
Yes, please. I hope it's possible.
Can you see my slides?
We are seeing your first slide, which says cancer stem cell hypothesis. Can you please speak to that?
Can you see this slide?
No, we don't see your slides.
Oh, really? Just a moment, please. Can you see the slides?
Yes, now we can see your slides.
Okay.
Yes,
Can I start from the slides?
please go ahead. Thank
Okay.
you.
Thank you. So, cancer stem cell hypothesis has been reported. Cancer cells are heterogeneous population, and cancer stem cells represent a small subset of cancer cells. Cancer stem cells are capable of providing progeny cancer cells and are considered to be related to resistance to conventional therapy. Lineage tracing is a powerful tool to identify cancer stem cells in vivo. Lineage tracing enables tracing cell fates by genetic marking using Cure-Lock-Spit system and reporter. For example, if gene X marks cancer stem cells, blue cancer stem cells provides descendant blue cells. If gene X does not mark cancer stem cell, it does not provide descendant cells, and blue cancer cells eventually disappear. As a candidate for a marker of pancreatic cancer stem cells, we focused on DCLK1. Immunostaining for DCLK1 shows DCLK1 is expressed in a very small subset of murine and human PDAC, as shown here. We first performed lineage tracing of DCLK1 positive panning cells in established panning. In panning PDAC. To this end, we used DCLK1 Cure-ERT knock-in mice. We performed lineage tracing of DCLK1 positive panning cells in established PDAC using a dual recombination system. In this system, panning was formed by flip-base FRT system, and lineage tracing was performed by Cure-Lock-Spit system upon tamoxifen injection. Before tamoxifen injection, all PDAC cells express tomato, whose color is red. After tamoxifen injection, DCLK1 positive PDAC cells and their descendant cells express GFP, whose color becomes green. If DCLK1 marks PDAC stem cells, PDAC cells will be replaced by descendant green GFP positive cells. So before tamoxifen injection, only a very small fraction of PDAC cells express GFP, which is the same expression pattern of DCLK1. 14 days after tamoxifen injection, GFP positive PDAC cells dramatically increase. This data indicates that DCLK1 positive PDAC cells provide number of descendant PDAC cells and increased over time. This data demonstrates that DCLK1 marks PDAC stem cells in vivo. Next, we perform lineage tracing of DCLK1 positive PDAC cells in liver metastasis. Before tamoxifen injection, only a small subset of metastatic PDAC cells express GFP1 in the liver, which was the same as the primary PDAC. 14 days after tamoxifen injection, GFP positive metastatic PDAC cells dramatically increased over time. This data indicates that DCLK1 positive PDAC cells provide descendant PDAC cells and increased over time in the liver metastasis as well. We next further performed live mouse lineage tracing of DCL1 positive PDAC cells in the same PDAC region in the same live mice. To make live mouse lineage tracing possible, we generated a glass window on the live mouse abdomen as shown here. Before tamoxifen injection, only a very small subset of PDAC cells expressed GFP as shown here. Uh, 7 days after tamoxifen injection, GFT positive PDAC cells dramatically increased over time in the same region in the same mouse. So this data clearly indicates that DCLK1 positive PDAC cells provide descendant PDAC cells in the same region. And this data demonstrates that DCLK1 marks PDAC stem cells in vivo. We next investigated spheroid xenograft formation ability of DCLK1 positive PDAC cells. To characterize DCLK1 positive PDAC stem cells, we next performed FACS sorting of DCLK1 positive PDAC cells in established PDAC. As shown here, DCLK1 positive PDAC cells have a high capacity of spheroid formation in vitro. In addition, DCLK1 positive PDAC cells have a high ability of xenograft tumor formation in vivo, as shown here. These data support that DCLK1 masks PDAC stem cells. We further investigated the gene expression profile of DCLK1 positive PDAC stem cells. Microarray analysis shows that genes involved EMT, stem cell genes, and invasiveness were upregulated in DCLK1 positive PDAC stem cells compared to DCLK1 negative PDAC stem cells. In summary, we could directly visualize PDAC stem cells by lineage tracing of DCLK1 positive cells in primary PDAC and metastasis in vivo. DCLK1 positive PDAC stem cells have a high capacity of spheroid tumor formation. This is acknowledgement slides. Thank you very much for your kind attention.
Thank you very much, Dr. Fukuda, for this talk. So I guess now the session is open for questions and answers. And I believe that both of the virtual speakers are also available to answer any questions. So please, when you get to the phone, do address who the question is for.
Yes,
Hi, Dr. Fukuda. We can hear you.
Yes. Yes. Yeah. This is Dr. Takaori from Nagahama. And I have a question for Dr. Wood. May I ask questions?
Yes, please go ahead.
Yes. Thank you, Laura, for the excellent and beautiful presentation. As you know, 10 to 30% of elder individuals have pannings, while only 0.02% of them have high-grade pannings, according to the previous autopsy studies. So the challenge is how we can identify those low-grade pannings, which with potential to progress to high-grade ones and to PDAC. So should we apply scoring system or cluster model to the genetic analysis and find out which panins are real precursors to PDAC? Or should we chase something like DCLK1, as Akisa has demonstrated that it is a stem cell marker in mouse PDAC? Or does 3D model help us identify how and which panins progress toward PDAC? Can you answer to this question?
Yeah, thank you, Dr. Takayori. I think that's a really key question. In our cohort, we did have, I think, 3 high-grade pannins. Each one was directly associated with a low-grade pannin. And I think one of the key future studies is really, in addition to the genetics, comparing the microenvironment and really seeing how is the high-grade pannin different from the immediately adjacent low-grade pannin, and also other low-grade pannins in that same pancreas. So we can identify both features of the high-grade pannin and also the low-grade that gave rise to that high-grade pannin. I think we'll need to enrich our cohort for high-grade pannin, because if you just blindly take normal pancreas, you'll get almost all low-grade. So I think accumulating a cohort of high-grade pannin to then model in 3D, find the associated low-grade, and then from there do all these studies is a key next step. And I think that any markers like DCLK1 or DLCK1, sorry, can be incorporated into the 3D models, and we can see kind of how they are distributed in human pancreas in 3D. So I think that the 3D approach has a lot of power to incorporate markers of interest into these models.
Yeah, thank you.
Thank you.
Hello. Okay. So my question is for Dr. Majumdar. First of all, thank you for that wonderful talk. And the question is, you mentioned like how much we eat and what we eat matters, but does it also matter how we store those fats? Because you showed some interesting data in male versus female mice. And again, in humans also the patterns of fat storages are different in men and women, visceral fat versus subcutaneous fat. Does that make a difference in the manner in which the fats play a role in promoting the PDAC pathogenesis?
That's a great question. It's been known for quite a while now that using lard-based high-fat diets in genetically predisposed pancreatic cancer models show differences between male mice versus female mice. So female mice don't gain as much weight, and they don't induce as much tumorigenesis. We don't know that for sure in our high oleic diets. We're exploring that now in our sort of broad panel, but I wouldn't be surprised given differences in adipocyte storage and release of free fatty acids that might play a role in the sex-specific differences that have been observed previously, and perhaps might also result in sex-specific differences with oleic acid as well. So that's an area that we're eager to explore.
Jason Pitaresi from UMass Medical School. I have a question for Dr. Vialli about the epigenetic memory in the epithelial tumors. Do you think this is something specific to the pancreas, or is this across all epithelial tumors? Might be a tough question to ask.
Honestly, I don't know. I speculate that in all tumor in which exists a universal driver, for instance, melanoma, or also GBM, there maybe the oncogene is important for coping with some sort of damage or process that actually is ongoing. About specifically epithelial memory and tumorigenesis, at the moment, I don't know. I think actually that is a common, a common process happening in other tissue, has been demonstrated in the skin, for instance, from Elaine Fuchs, that keratinocytes basically are able to acquire a memory of previous damage and repair faster, again, mediated by inflammation. And there is another paper from the group of Rando, I guess in 2019, that is the first description that when you damage muscles in the body, all the stem cells that generate muscles around the body get activated. At the time, they were not able to identify any mediator, but and they describe a new phase of cell cycle, a new state that actually G0 alert. So it looks like that actually if I damage a limb or a leg, the other one get activated, and those cells are more prone to repair, enter the cell cycle and repair the tissue. So for sure, I mean, there is a response to damage that is mediated probably, I guess, there are mediators that probably are cytokines or other things. And then again, probably there is a reprogramming. So I think actually is a general mechanism as being just described in the skin, pancreas, and muscle cells. But and I mean, I think actually, to my knowledge, our work is just the only one connecting this reprogramming with the tumorigenesis. So at the end, really, we think it actually is a a new epigenetic state that makes cells, prime to be so to be really transformed by oncogenes. So we have separation of different checkpoints there, for instance, senescence. And I mean, they are just more prone really to be transformed. In mouse, it lasts for several months. We test at least 3 months that actually in human correspond probably to 10 years. Obviously, it's difficult to understand if actually in humans is the same. But again, so in mouse, it happens.
Do you think there are KRAS mutations in histologically normal acinar cells?
This is something that we study, and I wanted to ask you the same question because I mean, obviously we study mouse, right? I know it happens. We are using digital PCR to detect them. The frequency is very low. My question is for you. What is happening if you sequence the rest of the tissue?
Yeah, we haven't done it yet, but we're working on it to see if you take histologically normal acinar cells or ductal cells, if you can find KRAS.
That is interesting also, maybe in light with all the work, about somatic clonal expansion,
Yeah.
right? So basically, maybe you will find something there.
Hi, this is Shishmita. I'm from University of Nebraska. I have a question to Dr. Majumdar. So when you are exposing your animal models to the high-fat diet, so what do you expect? Whether you have shown the data for the tumorigenicity potentiality. So have you seen any fat accumulation to the pancreas? And that is the first question. And the second question is, if the acinar cell is getting substituted with the fatty acid, fat accumulation, not the ductal cells, so there is a chance of getting diabetes. So how it is directing the path for getting diabetes or the PDAC, whether genetic modulation is playing the role in that?
So we don't see fat replacement of the pancreas in the high-fat diets. We do see increased peripancreatic fat. That's similar across the high-fat diets, and the mice gain comparable amounts of weight on most of the high-fat diets. So that doesn't seem to be the only sort of driving force there. The diets have very different impacts on insulin resistance. It turns out that some of the diets that are most insulin resistance producing actually don't have the highest levels of tumor burden. So the sort of the biggest correlate that we did find in looking at physiologic parameters and dietary parameters was really the fatty acid component, and really oleic acid was the big driver.
Thank you.
Hi, Kevin Stavely O'Carroll from the University of Missouri. I have a question for Dr. Majumdar. If the mice are on a ketogenic diet with low carbs and are overfed oleic acid, do they still have the same risk?
It's a great question. We have not done that. So all of them are getting 45% kilocalories fat, but the carbs and the proteins are controlled at 35% and 20%. We haven't tried to alter that.
My question is to Dr. Mazunda. Sorry, cannot pronounce your name right. So in your cell paper, you said in a high-fat diet, and you induce the beta cell produce CCK. So my question is whether this producing CCK is only under the KRAS condition or even just regular obese mice also will do that.
Yeah, the CCK story was based on the genetic model of obesity, so the leptin deficient mice. We see CCK expression in islets in that model, irrespective of whether they have KRAS mutation. Also true in the leptin receptor deficient mice, the DB mice, with or without KRAS mutation, you'll get islet CCK. And in the high-fat diets, if you feed long enough, you can get some CCK expression as well. So it seems to be independent of KRAS mutation.
With that, if you have a pancreas produce CCK itself, will that cause the pancreatitis?
So it's possible. We haven't formally excluded that. We've used CCK transgenic animals that we can express CCK specifically in beta cells to comparable levels found in the genetic obesity models. And in those models, you don't see pancreatitis until the mice are beyond a year of age. When we see the tumor phenotypes in that context, we see them within months. So we think that it may be playing a role, inflammation or pancreatitis may be playing a role, but it may also be sort of a direct effect of CCK and altering acinar cell identity as well, independent of the pro-inflammatory effect.
Thank you.
I just wanted to pick up on something that Laura said about looking at pannins that, advanced grade pannins next to early grade pannins. And I was wondering whether it's time to also look at the stromal cells around those pannins, because there's heterogeneity of stellate cells and some are thought to be restraining and then they turn into facilitatory cells. And I was wondering whether there's some signatures you might look at for those.
Yeah, I completely agree. I think that we started with the genomics in a tumor cell autonomous manner, but it's clear the microenvironment plays a really important role in knowing, I'm not the right expert to talk about all the CAF heterogeneity, but there's multiple different types. And I think doing profiling of the high grade and the low grade pannins and also low grade pannins that don't give rise to high grade and looking how the stroma is different is going to be another really important piece. So thank you for that.
Well, thank you so much. If there are no further questions, we'll close this session. And I would like to thank all the speakers and especially Dr. Wood for assisting with co-moderating this session because Dr. Takaori couldn't make it. And to all of you for attending and participating in this conversation. Thank you.
Thank you.
Thank you.
Thank you.